What if your next breakthrough medicine was locked inside a tiny vial, and its power depended on every single chemical bond? That's the reality of peptide research. These chains of amino acids may be small, but their role in science and medicine is huge. From cancer immunotherapy to metabolic studies, peptides drive discovery, but their impact depends entirely on how carefully they're made. Let's take a walk through the process, from chemical assembly to cold storage, and see why precision in manufacturing is the foundation of reliable research. Solid phase peptide synthesis. Modern peptide manufacturing relies on solid phase peptide synthesis. Imagine building with Lego blocks, where every piece must fit in exactly the right place. The process starts with the first amino acid anchored to a resin bead, which allows chemists to wash away byproducts while keeping the growing chain intact. Each amino acid comes with protective groups to prevent unwanted side reactions. After removing the protective group at the N-terminus, the next amino acid is coupled using activating reagents. The cycle of deprotection, coupling, and washing repeats until the sequence is complete. Finally, strong acids release the peptide from the resin and strip away side chain protections. What's left is a crude peptide, ready for purification. Purification, turning crude into clean. The crude product is a messy mix of incomplete chains, side products, and the desired peptide. To separate the signal from the noise, chemists turn to high-performance liquid chromatography, reverse phase. HPLC sorts peptides by hydrophobicity, producing a pure fraction that can be collected and analyzed. Purity is then confirmed with mass spectrometry, which verifies the peptide's identity by molecular weight. Analytical, high-performance liquid chromatography checks how much of the final product is actually present. These tests are more than technicalities. They guarantee researchers can trust their results, free from interference by contaminants. In peptide science, purity equals reproducibility. Lyophilization, freeze-drying for stability. Purified peptides are often still in solution, which isn't stable for long-term storage. The solution? Lyophilization or freeze-drying. The peptide is frozen and then placed under vacuum, so the ice sublimates directly into vapor, leaving behind a dry, stable powder. This fluffy cake is easy to ship, simple to weigh, and can last for months or years if handled properly. Lyophilization doesn't just extend shelf life, it preserves the peptide's structure and function so researchers can rely on it every time they open a vial. Handling and storage. Even freeze-dried peptides need protection. The gold standard is storage at -20 degrees Celsius, with -80 degrees Celsius preferred for sensitive or long peptides. Because moisture is a silent destroyer, vials are sealed tightly and often paired with desiccants. Many peptides also require light protection, which is why amber vials are commonly used. Once reconstituted in solution, peptides must be handled carefully. Repeated freeze-thaw cycles can degrade them, so scientists aliquot samples into small portions. Sterile handling is equally important to avoid contamination, especially in cell or animal studies. A single slip in storage can undo all the precision of synthesis and purification. Why precision matters. Why all this care for such tiny molecules? Because quality determines reliability. Impure peptides can yield misleading results, waste resources, or cause unexpected biological effects. Poor reproducibility is not just an inconvenience, it's a setback for global science. On the other hand, precisely manufactured peptides provide consistency across labs, enabling breakthroughs that scale from benchtop studies to clinical applications. This reliability is critical today as peptides expand into medicine. They're now central to vaccines, cancer therapies, and metabolic treatments like GLP-1 analogs. Regulatory bodies such as the FDA and EMA require proof of purity, stability, and identity before peptides move into trials. In short, manufacturing rigor translates directly into research credibility and patient safety. Advances in modern manufacturing. Peptide chemistry keeps evolving. Microwave-assisted SPPS shortens reaction times and boosts yields, especially for long or tricky sequences. New resins improve solubility and reduce aggregation, while advanced coupling reagents minimize side reactions and stereochemical errors. Analytical tools are sharper too. Mass spectrometry can detect impurities at trace levels, while stability testing predicts shelf life under stress conditions like heat, humidity, or light. Even packaging has advanced, with amber glass, inert sealing, and temperature monitors during shipping. Together, these innovations mean researchers today can access peptides that are purer, more stable, and more reproducible than ever before. Illuminated Peptides. At Illuminated Peptides, these high standards are not optional, they're our foundation. We partner only with verified suppliers of amino acids, resins, and reagents. Our sourcing facilities are inspected, and our quality control spans every step, from synthesis to final packaging. Every peptide is verified by mass spectrometry and high-performance liquid chromatography before reaching researchers. Storage and shipping follow strict protocols, with freeze-dried peptides sealed in amber vials, protected from light and moisture, and transported via cold chain logistics. By combining rigorous science with responsible supply chains, we deliver peptides researchers can trust to perform exactly as intended. The journey of a peptide, from resin-bound chain to cold-stored vial, is one of precision and care. Every step ensures these molecules remain reliable tools for discovery. In a world where reproducibility is everything, high-quality peptides aren't optional, they're essential. At Illuminated Peptides, powered by OmniNext Tech, we combine science and technology to light the path forward in peptide research. Trusted, transparent, illuminated, America's #1 choice for research peptides. These products are for research use only, not for human use. Enjoyed this? Like, share, and subscribe to stay connected with the latest in peptide science.